In this lesson we're going to look at chromosomes, and this is basically a form that DNA takes whenever it's going to get to the point where we need to divide cells. So we're going to talk about cell division later on in this module, but just to give you a little bit of background, a chromosome is just condensed DNA that's coiled around proteins. When it's not condensed, that DNA is called chromatin, and the chromosome has two halves that are called chromatids. So these names all look very similar to each other. We'll have to go through and kind of look at how these things look, and then that way hopefully you'll remember the name with the image in your mind. So here's an example. This is a chromosome. They form during cell division. Here you can see this is the nucleus inside of a cell, and all these little looks like X's are the chromosomes. As I have labeled here, each half of the chromosome is called a sister chromatid. So you have this half down here where we have it already highlighted in pink, and this half over here is another sister chromatid. They're held together here in the middle by what we call a centromere. And the coiled DNA you can see here. So if we just take a look at this, here's the DNA that we know. It coils up around these proteins and forms this much more condensed version of DNA called a chromosome. A chromosome can either be one strand of DNA or two. This would be, the two strand would be before the division process, and then this one-stranded chromosome would be after the division process. And it's not going to stay like that for very long. It'll uncoil back into its chromatin. So a cell may actually look like this, or like this one over here. Both of them are said to have two chromosomes. Again, this would be after division, and this one is going to be before, because we have the two halves of the chromosome still there after and before division. So again, here we have the sister chromatids. So one side's, I have it in pink, one side's in green. Then the centromere is right here, holds the chromatids together. And then this is what we call a karyotype. This is showing you the chromosomes all lined up. So these would be human chromosomes. There are 23 pairs, which gives you a total of 46 total chromosomes. You get 23 from your mom, and you get 23 from your dad. That's what the M stands for. So that would be mom, dad, mom, dad, dad, mom. It doesn't really matter what order they're in. And you can see we go all the way down until we get to the 23rd set, which is the sex chromosomes. And if you're a male, you have an X and you have a Y. If this were a female, you'd see two X's there. Something we'll talk about a little bit later on. Lastly, I just want to sort of give you an idea of what chromatin looks like. Remember, this is the uncoiled DNA. So if I have a cell, and inside the nucleus, this is a cell nucleus, and then just change colors here. My chromatin's going to look kind of like spaghetti. So it's all stringy and uncoiled. This would be really hard to separate in the division process. So that's why it condenses into chromosomes, and you'll learn that later as well.